attendance. This is our 4th webinar since we began the series in October of 2018. Next slide, please. So I'm Meg Whitaker. I'm the A4 Program Committee Co-Chair with Dr. Lauren Hine, and the focus of our webinar today is how to get to safer using non-animal testing methods. And we've really got 3 goals that we'd like to communicate and discuss today. We want to present what are new approach methods, better known as NAMs. Specifically, why are they of interest and how are they being used in practice? We'll discuss considerations for using NAMs in the context of conducting alternatives assessments. And finally, where can we learn more about NAMs if you're interested? Next slide. So we've got today a great group of speakers and 2 esteemed respondents. All of these individuals have so much experience focusing their careers on identifying safer chemicals, materials, and products. Dr. Pam Spencer is Senior Director of Regulatory Product Stewardship and Quality at Angus Chemical Company, and we're also lucky to have her as A4's inaugural president. Very brave, Pam. Dr. Amy Klippinger is the President of the PETA International Science Consortium Limited. In her role within the Science Consortium, she collaborates with industry, academia, and regulatory agencies to promote robust non-animal toxicity testing approaches. So Pam and Amy are our 2 presenters. And then today we're mixing it up a little bit. We've got 2 respondents. We've got Dr. Lauren Hine, who's the Director of Safer Materials and Data Integrity for ChemForward. Dr. Hine's career has been spent working to protect human and environmental health by applying alternatives assessment, green chemistry, green engineering, and multi-stakeholder collaboration to the development of products and processes. She's one of my favorite engineers. And we have Sherry Franjevic, who's a Green Screen Program Manager at Clean Production Action. Over the last many years, she supported the evolution and growth of the Green Screen hazard assessment method, helping clients transition towards the use of safer chemicals. So we've got an action-packed next hour. Next slide, please. So let's just spend 30 seconds going over webinar logistics. Due to the number of participants on the webinar, and this is the largest webinar to date, all lines will be muted. But we want to hear from you. So if you'd like to ask a question, please type your question in the Q&A box located in the drop-down control panel at the top of the screen. We'll have questions at the end of the presentations. Questions will be answered, as I said, at the end of the panel discussion. The webinar is being recorded and will be posted on the slide deck on the A4 website, just like the previous webinars. And at the end of the webinar, we will launch a short evaluation survey to help us with future webinars. So we want to know what you need, what you like, what you didn't like, and we'll use that to keep making better and better webinars. So with that, let's go to the next slide. And I'm very happy to introduce Pam Spencer, who's going to be providing you with an introduction to NAMs and application of NAMs in new product innovation. So take it away, Pam.
Thanks very much, Megan. Thanks to everybody for participating today. What I'm hoping to do today is really just kind of level set for the group so that we're all speaking the same language when we're talking about what NAMs are. Go into a little bit about the types of NAMs, because I think it's a broader definition than what maybe some people might think. And then just talk about a couple of areas. One, why is there so much, activity around NAMs? Aren't the animal tests that we've been using good enough, and really aren't they the gold standard? And then really go into a couple of examples that can highlight how these are actually contributing to product innovation and in some forms helping us make decisions about advancing more sustainable alternatives. So next slide, please. So what are NAMs? It stands for New Approached Methodologies, but it has a lot of synonyms that get associated with it. So if you hear predictive toxicology, if you hear toxicity testing in the 21st century, Tox 21, non-animal alternatives, alternative test methods, these are all terms that represent this new direction that we're taking with the field of toxicology. And the term was really coined by the National Institute of Environmental Health and Safety when they released their strategic roadmap. And it was really meant to be adopted as a broadly descriptive reference to any non-animal technology, methodology, approach, or combination of those that could provide information on chemical hazard and risk assessment. And I emphasize the point information because they really moved away from data requirements to say, what information do we need to make better decisions and to better characterize the health and environmental impact of our chemicals that we're assessing. So just want to keep that front and forward, that we're really talking about information needs. Next slide. So NAMs come in many shapes and sizes. You could think of them as tools, you know, in silico or computational tools where you have long-standing tools that we've used like quantitative structure activity relationships, QSARs, but also moving into more high-tech and newer scientific landscapes where we're looking at machine learning and big data and how can we build more informative models leveraging this big data, not just to understand hazard characterization, but also as it applies to exposure and high throughput exposure modeling. And then you can think of some of the other more compact, I like to call them assays, where we're talking about in vitro biological profiling, where we're using surrogates of biological systems to interrogate for different biological activities. And these could be cell cultures, they could be 2, 3D organotypic cell cultures, where you actually have multiple cell types that would represent a target tissue in a liver or a lung, say, but they can also include things like genomics and transcriptomics and looking at profiling to see how fingerprints of different chemicals compare, all the way to the newest and greatest thing, organs on a chip, where with microfluidics and dynamics, we're actually starting to build miniaturized organ systems and mini, almost human on a chip type of assays that can be used on a very micro level. But in addition to the tools, there's frameworks, and frameworks are also part of the NAMS toolbox. And these are really ways that we go about to make decisions. So you can think about adverse outcome pathways, defined approaches, integrated approaches to testing and assessment, or IATA. So these are all tools or all approaches that are encompassed within the term NAMS. Next slide. So I want to just get a little bit more into the details on frameworks and to show how these build off of one another. So if we can go to the next slide. So adverse outcome pathway. So an adverse outcome pathway is a model that identifies the actual sequence of biological events that are required to produce a toxic effect in an organism once it's been exposed to the chemical. And as an example here, you see a skin sensitization AOP, because it's probably one of the most better characterized ones. And what you see is that you have chemical structure properties that feed into that. You have a molecular initiating event, and then you have a series of biochemical events that occur at the cellular level, then at the organ level, until ultimately you get the adverse outcome, which is what you would see as a skin rash or an allergic reaction. And these AOPs are very important because they not only give us those biochemical events that occur in the pathway, but they also allow now assay developers to say, okay, can we develop assays that could actually query these different key components in the pathway from exposure to the adverse event. And OECD has been promoting this, and I've given you the link to the AOP wiki. So there's a wiki with many different AOPs, and it's always building. And this is where OECD is supporting the development of these adverse outcome pathways, keeping a catalog of them, and anybody can go out and see them. And again, as I said, this helps assay developers identify how they can, you know, leverage these pathways to be able to look at these key events in the biochemical adverse outcome pathway. So the next slide. The next slide is defined approaches to testing. So you can think of this as a framework. Now that we have an adverse outcome pathway, is there a way now that we can combine the information from the various key events to drive a decision? And so again, if we look at the adverse outcome pathway for skin sensitization, you can see that there's different key events, and now not only have the assays been developed for those key events, but we actually have OECD test guidelines that we can use to measure for those key events. So covalent interaction with skin proteins, there's a guideline for that. Induction of inflammatory cytokines and surface molecules, a guideline for that. Now we can start to take these information and put them into a flow diagram where we can say, okay, if we do 2 out of 3 of these key events and they're consistent, both positive or both negative, then we can use those 2 concordant results to say we can classify now either the chemical as a skin sensitizer or not. And so there's a number of defined approaches that are under development. This is one that has been fully vetted and is now able to be used in registration, and Amy will probably talk a little bit more about the registration piece, but these are now in place to use so that we can replace the animal testing with looking at these biochemical events and making a determination about whether or not we have a skin sensitizer. And I've given you the link to some of the defined approaches that NICEDM has put in place, and you can go out to that link and see the other defined approaches that they've developed. Next. Now, defined approaches and other information can actually be used in a more structured approach called integrated approach to testing and assessment. And if you were in attendance at the last symposium for the Alternative Assessment Symposium, we presented some of this work on how IATAs could be used to look to see if we have enough data or information in a weight of evidence format and make a determination on whether it's sufficient to classify that endpoint or whether it would drive us to do further testing. And so then again, this is kind of think of it as building lines of evidence. I think of it like for a court case that you're bringing all your streams of information in and saying through that weight of evidence, do I have enough to say with any degree of certainty that I can classify that endpoint? If not, then what data do I need or what information do I need to be able to do that? And that would then guide the testing that would be driven. So rather than just checking a box and saying I've done all of this testing, this is a more integrated approach that's driving testing based on the information need. And again, another good framework that represents how NAMs are being used. Next slide. So why NAMs? I know there's a lot of concern sometimes that, we're using the animal tests, they've served us well, they've been good indicators, and aren't we just using NAMs to really find a quicker, cheaper way of not doing the testing? Well, NAMs addresses three, I would say, really important challenges in the chemical safety assessment world. One, yes, they are quicker, they're lower cost, but the point is that we can actually now evaluate many, many more chemicals than we could with the old animal test methods, which some tests take years to complete. It's just not feasible with all the chemicals that are on the market to use that kind of a tool to really conduct our safety assessments. We need something that fits the magnitude of the chemicals that we have to assess. And Amy will get into this a little bit more in her presentation. But the other really important thing is that they now provide us with human relevant information for industrial chemicals, where we could never have testing in humans before. Now we can use human relevant cell lines and even organs on a chip to begin to say, what's the relevance and the impact to human biology? And then the third component in the area that I really get excited about, besides the human relevance, is that now we can screen for chemicals much earlier in product development. And if you have an understanding of how product development takes place, usually with animal tests, you really can't get to test a chemical in an animal till you've scaled up production, at least to the pilot plant level, because you need a certain amount of chemical to test. But now we can start to screen very early, even before a research chemist makes that material at the bench. And that's a really exciting thing to advancing more sustainable chemistries into the marketplace. Next. So they offer us really good things, but are they telling us the same things as the animal tests? That aren't the animal tests the gold standard? And if you look again at the skin sensitization model and applying a defined approach methodology, you can actually begin to do computational assessments to look at combining different in vitro data streams and in silico data streams and seeing how well they predict the outcome of the chemical and compare that back to the animal test to say, okay, do they do a good job of predicting or not? And the skin sensitization data is really interesting because this is really one area where we do have human data as well. And so that's exactly what Nicedum did, is they said, well, let's take a look. How well do these new methods predict based on the results of the animal test and what we know about the human data that we have? So if we go to the next slide, and what we find is that NAMs actually outperform the animal test for predicting skin sensitization. So they looked at combinations of machine learning, computational methods, a combination of computational methods and in vitro methods, and looked at the predictive outcome. And if you look at the hazard prediction, the mouse predicted the chemical correctly 74% of the time. Defined approaches actually were better, 75 to 85% of the time. And then if we look at potency, so the class that the sensitizer falls in, the mouse was only about 59% and the defined approaches were similar. So I've given you the reference to the publication, which was by Kleinstrauer et al. That really talks about this. But what we're starting to see is that as we look at the animal data that we have and we compare it to the outcome of the defined approaches, these NAMs are performing as well, if not better, than the animal test in almost all cases. And so I'd encourage you to go look at that literature. But it does beg the question, as we're trying to validate these tests, what's the right benchmark to validate against? Is it really the animal test, and is the animal test really the gold standard that we should be comparing them back to? Maybe it's something in between, and where we have this human data, we need to bring it in and use that to help build the confidence and the validation that we're doing for these. All right, next. So now I'm just going to briefly hit on two examples of how NAMs can be used and the way that we're using them today as we're looking at new product development. So one challenge here, and I'll be brief on this, is that we have a commercial product that's registered, and it's an isomeric mixture 50-50, and that's what's registered. But we'd like to have the flexibility to be able to vary the isomer ratio in the product. But in order to do that, it would require that we would have to register each isomer individually, because the way the regulations work, it doesn't give you the flexibility to vary those isomers. So what we registered is 50-50. That's our guidelines that we have to stick to. So obviously, we don't want to go through and generate testing in animals on each isomer separately. We want to be able to read across. And is there a way that we can enhance our confidence that reading across to the isomers would adequately represent the biological activity of the racemic mixture for which we have all of the animal test data? So you can use a transcriptomic approach to do this, and you can actually treat cells, and usually you're going to treat cells with the target tissue of interest, and then you can gather the RNA and look at the transcriptome for each isomer to see which biological pathways it is stimulating and to see how similar they are. And this is just an example because we're just in the process of doing this work right now. But philosophically, what you would do is you would look at the overlap of the genes that are being activated, either upregulated or downregulated, and from that you can tell with a certain degree of confidence that the mode of action is going to be either similar or different. And so we're using this to help us have more confidence that we can read across and that we don't need to do additional animal testing on the separate isomers. So that's one example, and I know they're using this a lot also in the cosmetic industry where they can't do animal testing. They're actually using this to look at hazard outcomes. Next slide. And then the other application that I talked about is in new product development. So here's an example where we have a candidate that we want to replace. It's a more toxic candidate, and the chemist says, you know, gee, I've got some ideas on how I might develop a structural analog that we think will be not only better in performance, but lower in toxicity. So they can actually give me these structures, and I can convert those structures to SMILE codes. And then I can use various predictive models, and we typically use around 3, 4, 5 different models, and I've listed the models on the right hand of the slide that were used in this assessment. And we can start to generate what I call this little bit of a heat map to say what candidates look better just from a computational screen. And I can now help guide the chemist resources in a direction towards molecules that we think are going to truly have a better human health and safety profile. So if you look at this particular screen, you can see that it looks like F, just based on the heat map, looks like the best candidate. It doesn't seem to have all of, you know, some of the eye irritation and reproductive endpoints that might be of concern. We can look at B and C and say, yeah, they're kind of in the middle. But when we look at D and E, we would just simply tell the chemist, don't waste your time on those 2 molecules. There's too many structural alerts. Let's abandon those. And now we can then move a few potential candidates forward that we can move into an in vitro test system to sort of truth the computational results that we've gotten in this very early screen. But it's really critical for allocating resources and making sure that we're investing our time and money in products that are truly going to be sustainable. So these tools help amazingly in that way, and we're using them very often as we're looking at new chemical candidates. Now, I just want to make everybody aware that doesn't mean that we won't be doing animal testing ultimately to register a new product. That's required, but what it does do is help us guide us to use animal testing in the most efficient way, in the way that's going to give us the most valuable information as we go forward. Next slide. So I'm going to just stop there and just leave everybody with some food for thought as A4 members and as people that are looking at alternatives. The field of NAMs is advancing really rapidly, and so, you know, how can NAMs be used in our field of alternative assessments? And if we think about how we can use them, is there a role that we can play in helping to establish confidence in the way that these are used? And if so, which NAMs and how? And should we be advocating or offering training for our members? Training comes up time and time again in every, I'd say, sector that we're talking about, regulatory to industry. And so what should we be doing in that area? And are there other opportunities? And then I'll just leave you all with just a plug, is that we have started a new SOT specialty section called Sustainable Chemicals Through Contemporary Toxicology. This is going to be a pointed partnership between A4 and SOT, and it's really going to be a forum to highlight these examples like I've shown today and to have these discussions. So for those of you that are SOT members, I would encourage you to check that out at the annual meeting. And with that, Meg, I'll turn it back to you.
Thank you, Pam. That was really a great presentation and a beautiful overview. So lots of food for thought, and I'm sure there'll be a lot of questions. And there will be a luncheon for the SCCT on Monday at SOT. You can see it in the program, so I hope some of you on the webinar today will join us for lunch. Now we're going to hear from Amy, who's going to be presenting and discussing the development and use of predictive animal-free toxicology testing approaches. So Amy, hand the baton to you.
Great, thanks, Meg. So for those of you who may not be familiar with my organization, we were incorporated in 2012 and we're made up of about 20 scientists spread throughout the US, UK, Germany, Belgium, and India at the various PETA affiliates. And we focus on advancing the implementation and regulatory acceptance of robust animal-free testing approaches that protect human health and the environment. And we do this through various funding and training activities, as well as other events. Next slide, please. So my talk today will provide a high-level overview of why we're interested in NAMs, and I'll keep this very brief because Pam's already done a really nice job of talking about this. And then I'll present 2 case study examples highlighting how new approaches for respiratory toxicity are being developed and vetted. And then I'll end by providing resources on where you can find out more. Next slide. So for the past 15 years or so, there have been significant resources dedicated to advancing the development of non-animal testing approaches, which has been driven by a desire for better protection of human health and the environment, as well as ethical, time, and monetary considerations. The 2007 National Academy of Sciences report called Toxicity Testing in the 21st Century, A Vision and a Strategy was co-authored by industry, academia, government agencies, and NGOs, and really paved the way for a paradigm shift in toxicology testing to a system primarily based on human cells and mechanisms in order to more efficiently generate more robust data on human risks. So in the 13 years since this report was published, there's been a groundswell of support for optimizing and using these methods, including several commitments as well as legislation implemented by international regulatory agencies. For example, there are 2 strategic roadmaps that were published in the past several years that acknowledge the scientific breakthroughs that have generated new tools and methods that can be used to advance the field of toxicology. And these documents provide a strategy for how to translate these breakthroughs into a more predictive and efficient system for assessing the effects of chemicals. One of these roadmaps came out of the FDA and the other from the Interagency Coordinating Committee for the Validation of Alternative Methods, or ICVAM. For those of you who aren't familiar with ICVAM, it's made up of the 16 US federal agencies that generate, require, use, or disseminate toxicology testing information. So groups like the Consumer Product Safety Commission, US Department of Agriculture, Department of Defense, EPA, FDA, and so forth. I encourage anyone who hasn't seen these documents to take a look to see how various groups are working to collaboratively and transparently gain confidence in new methods and make progress in this space. Next slide, please. So I'm going to dive right into the case studies. There are endpoints where non-animal methods are quite well established. For example, methods that have been incorporated into OECD test guidelines and IATAs, and endpoints for which adverse outcome pathways have been developed. And Pam mentioned skin sensitization testing as a great example. However, today I'm sharing 2 case studies for an endpoint that is, relatively speaking, in the earlier stages because I want to highlight the process that these methods undergo as they're being developed. So why are NAMs for respiratory toxicology important? Inhalation is a major route of human exposure to substances. Therefore, it's important to characterize the risks or benefits that they may present. These tests are generally conducted in animals. However, differences in anatomy and physiology between the rodent and the human respiratory tract limit the precision with which the rodent test predicts a human response. For example, there are differences in the ventilation rates and breathing modes. Humans are oral nasal breathers, while rodents are obligate nose breathers. Anatomically, there are differences in the airway architecture and branching pattern of the respiratory tract. As shown in the figure in the bottom left-hand corner, the branching pattern in rodents is asymmetric or monopodial, which results in a relatively unimpeded airflow, whereas the branching pattern in humans, shown in the upper right, is bi or tripodial, resulting in an airway pattern more susceptible to deposition as bifurcation points. There are also species-specific differences in the cell types and their distribution within the different regions of the respiratory tract, differences in mucus composition, and metabolic activity of critical enzymes. And all of these differences affect the deposition of an inhaled agent as well as its clearance or retention. Therefore, there have been significant resources dedicated to developing human cell-based approaches that can characterize potential hazards of inhaled substances while also providing a better understanding of how an inhaled substance exerts its toxic effects in humans. So in this respiratory toxicology space, we have a real opportunity to move away from black box animal tests that became test guidelines about 40 years ago and were grandfathered in, even though they were never validated for their relevance to humans and never validated for their sensitivity and specificity. Next slide. So Pam already mentioned the varying complexity of in vitro systems, and for respiratory toxicology, these models may include your simpler cell lines or primary cells grown under submerged conditions or at the air-liquid interface. Also commonly used today are three-dimensional reconstructed human respiratory tissue models, which are commercially available from companies such as MatTech or Epithelix, and which represent different regions of the respiratory tract. There are also microfluidic lung chips. For example, the company Alveolex in Switzerland makes a human lung chip. Human precision cut lung slices are another model, and organizations such as the Institute for In Vitro Sciences are conducting contract testing using these precision cut lung slices, as well as other test systems. So there are various available models that vary in complexity and their advantages and disadvantages with regard to cost, length and culture, physiological relevance, and so forth. And the test substance and the purpose of the study will guide selection of the most appropriate system to use. The projects that are underway today are focused on further understanding these test systems and conducting systematic testing to show the advantages and limitations of each for specific testing purposes. Next slide, please. So the first example I'll give today is the result of a public workshop that we co-organized in 2016 with the U.S. National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, also known as NICETM. The goal of this workshop, which was attended by about 40 experts from regulatory agencies, academia, NGOs, and industry, was to better understand the state of the science and to identify gaps as well as the necessary resources and specific steps needed to overcome these gaps. Having a workshop and 2 open access peer-reviewed publications was important to ensure that anyone who wanted to be included in the discussions would know about the effort and could get involved. One workshop recommendation was to present specific case studies showing how these NAMs could be used. So in the months following the workshop, we had many discussions with experts in the field to discuss the experimental design, and then we began funding at VITO, which is a research organization in Belgium, to conduct this testing. Next slide, please. In this project, we're testing pairs of industrial chemicals that are similar in structure to see if our in vitro system can detect the difference in toxicity that's expected based on structural differences. And this is important because if we know that a test chemical is more or less toxic than a well-studied control chemical, that provides a gauge of toxicity. We also selected these test chemicals because we have a wealth of existing information on their phys-chem properties, their toxicity, and context of use. So we know generally the range of what to expect. We started by exposing B2B cells, which is a human bronchial epithelial cell line, to the test chemical at the air-liquid interface using a VitraCell in vitro inhalation exposure module. And I don't have time today to get into the detailed results, but preliminary data show a decrease in cell viability and an increase in cytotoxicity and in various inflammatory markers following exposure to increasing concentrations of the test chemical. So now we're testing the additional chemicals and positive and negative control chemicals in these 2B cells, as well as other 2-dimensional and potentially 3-dimensional models. And while I won't have time to discuss the experimental details today, we will be presenting these results over the coming year, including at the Annual Society of Toxicology meeting in March and at the World Congress on Alternatives in Animal Use in the Life Sciences in August. So if you're interested in learning more about this work, please look for us there. Next slide, please. You can just jump right to the next one. The second example is one that was discussed at a public December 2018 EPA FIFRA Scientific Advisory Panel meeting. This approach was submitted by the company Syngenta, and it was to assess the inhalation risk for point of contact toxicity of one of their chemicals. The approach was conducted as an alternative to a 90-day rat inhalation toxicity test for the re-registration of this chemical, meaning this is a substance with existing data and it's already known to be an irritating aerosol. So again, no time for the details today, but very simply, Syngenta's approach involved agrochemical workers wearing an OSHA versatile sampler tube to understand exposure and particle size distribution, and then using computational fluid dynamic modeling to predict the deposition of inhaled particles within a human respiratory tract, and then using Epithelix mucil air, their three-dimensional model, to assess various biological endpoints. And underpinning the strategy is an understanding of the mechanism through which the chemical exerts its effects. And while I can't do the experiment justice with such a brief summary, if you're interested in more information, you can read the SAP report or the series of detailed publications that are expected to come out soon. And coincidentally, just about an hour ago, the Society of Toxicology In Vitro and Alternative Methods Specialty section held a webinar specifically on this approach. So if you did miss it, you'll be able to view a recording of the webinar online soon. And this is also a good time to mention that a lot of time and thought from Syngenta, the contract research organization, they used the EPA, the scientific advisory panel, and other stakeholders went into this approach. And it's been a very transparent process that actually took years to develop and more time and money than it would have taken to just default to the 90-day rat test. So while an approach like this may likely be faster and less expensive in the long term, once it's established, there is an initial investment to develop and validate a new approach. Next slide. So I hope these case studies showed that we're not talking about just a one-to-one replacement or a one-size-fits-all approach. The optimal testing strategy will depend on the test substance, its context of use, and the purpose of the study. But these efforts do have common both technical and practical themes, such as the use of lung cells grown at the air-liquid interface, as well as transparency and involving various groups and expertise to develop, understand, and gain confidence in a new approach. Results from the ongoing studies I discussed today, as well as many others that are underway, are providing important additional information that will further advance the field. Next slide, please. Okay, so I hope after hearing my and Pam's talks, you're at least somewhat interested in learning more about NAMs and how they might be applied to your work. So the next question is, what resources exist to help you find out more? In 2018, the EPA published its strategic plan to promote the development and implementation of alternative test methods within the TSCA program. And this strategic plan includes a list of NAMs, which is a good place to start. The EPA notes that this list is not meant to be an exhaustive list, and it encourages stakeholders to consult with the agency early on in the development process, so to ensure that the information generated will satisfy the information needs. NAMs are considered in a fit-for-purpose approach because a particular method may be suitable for one regulatory decision context, such as prioritization or as part of a weight of evidence approach, but not for others, such as quantification of hazard or risk. This concept also holds true for other agencies. For example, the FDA will consider proposed NAMs, and its Center for Devices even has a formal program for companies to develop new methods in collaboration with the agency. Then let's see, this EPA website also includes more information about the criteria EPA uses to determine if a NAM is of equivalent or superior quality to an existing approach, and some guidance on whether it can be used in a specific context. Next slide, please. In addition to EPA's list, NICEDM also maintains a list of NAMs on their website. They also have a listserv called NICEDM News that, in my opinion, is, if not the best, one of the best sources of information on developments in the NAM space. If you go to the upper right-hand corner, I highlighted it with a yellow box there at the top, and click on support, you'll get to the page where you can sign up for NICEDM News. And also from this page, highlighted in yellow on the right side there, you can find out more about ICFAM, including the strategic roadmap that I mentioned earlier, and information about ICFAM's annual public forum, which is another great event and source of information. Next slide. My organization also maintains a list of NAMs, and we also have detailed endpoint-specific fact sheets listing OECD-approved test methods, how they work, their applicability domains, and which GHS categories they're able to delineate. Next slide. We also keep a list of training videos and webinars on our website, and a list of companies that offer in vitro testing services. You can go to the next slide. And we organize free in-person laboratory training for researchers interested in using non-animal methods, because we know that an understanding of what methods exist, how to conduct them, and how to interpret the results from these assays is critical to adopting non-animal approaches. So for these sessions, we've sponsored groups like the Institute for In Vitro Sciences or the Laboratory of Mathematical Chemistry to provide hands-on training on in vitro and in silico models. There are also other groups that provide these types of trainings. There's a great continuing education program called NURA, which stands for NAM Use for Regulatory Application. And Pam was actually one of the presenters at the last NURA training. We also provide travel awards for scientific conferences and courses like the Institute for In Vitro Sciences annual Practical Methods for In Vitro Toxicology workshop, the European Commission's Joint Research Center Summer School, and the Summer School at Johns Hopkins on Innovative Science Without Animals. So while this isn't an exhaustive summary, it shows the types of opportunities that are available. And I realize I went through that pretty quickly, so if you didn't have time to copy down the links, or if you have questions or would like more information, I'm happy to connect, and my email address is shown on this slide. Thank you.
Wow, that's a really great presentation, Amy, and so many resources, because that's a question I'm often asked: where do I go? How do I start? So now we're going to hear from Amy, or Lauren and Sherry. They actually put a lot of the assessments into practice, and we want to hear your reflections and insights, Sherry and Lauren, particularly in light of making hazard-based decisions using NAMs. So I'll just pass the baton to you, and let's hear from you. I believe, Lauren, you're going to go first, correct?
I think I was going to go first.
You're going to go first? Okay. Oh, all right. Go ahead, Sherry.
Either way. Well, thank you, Meg and Pam and Amy, for this very, very rich, dense content. I think your expertise is just really phenomenal in this area, and there's so much that people can dig into. So thank you for your presentations, both as an overview as well as how to learn more. I'm coming at this conversation from the perspective of someone who designs hazard assessment methodologies that can be used as and incorporated into an overall alternatives assessment for identifying safer chemicals to replace toxic chemicals. And the tool that my organization, Clean Production Action, has is the Green Screen for Safer Chemicals, which has been around since 2007 and has been integrated into a lot of organizations' decision-making processes, particularly in the electronic sector, the apparel sector, and the building sector, as well as eco-labels in those sectors, including things like EPEAT and the U.S. Green Building Council's LEED standard, as well as companies such as Hewlett-Packard, Apple, and others, Levi Strauss, et cetera. You know, in terms of Green Screen, we have always had an inclusion of, you know, looking at all available data and information when doing a hazard assessment. And, the intention of green screen is to define a standardized way of evaluating chemicals such that you can compare them, and you can compare the results of different assessors, in particular licensed profilers or toxicology firms that do these assessments. And while we incorporate various forms of NAMs into the hazard assessment method, we have two pieces. One is to classify the hazard, and the other is to identify the confidence level. And this data tends to still be considered sort of lower confidence data in large part. But we do incorporate things like QSAR and silico, computer modeling, in vitro testing, et cetera. I think one of the places that, we see a lot of lack of measured data or animal data, or the data gaps that we see oftentimes, there's a few hazard endpoints out of the 18 that we look at. Neurotoxicity, endocrine activity, and respiratory sensitization are some of the most common data gaps. So one of the questions that arises for me, is how can some of this new data be used to start to fill in those data gaps? And we are doing a lot of work on, refining the criteria for endocrine activity to incorporate some of these different methodologies and frameworks. But I think that's a place where we're really, thinking about this. And then when I think about, needs and challenges from the perspective of a standardized hazard assessment methodology, one of the things that we like to do is point to authoritative sources for information on data interpretation. And right now, some of the key authoritative sources we point to are GHS, the Globally Harmonized System of Classification and Labeling of Chemicals, as well as some of the EPA risk assessment guidelines. And I noticed that oftentimes they're still heavily weighting animal data in their classification. So, one of the questions or needs that comes to mind for me is how do we identify some of those authoritative sources for interpreting data and using that for classification of hazard. And I think the other thing that arises that speaks to this similar point is just a need for consistency among assessors. So we have a standardized method, and for it to be valuable, we really need different assessors to come to the same conclusion. And so, how do we provide sufficient guidance in some of these areas, especially of emerging science, such that, there's sufficient information on interpreting those results to come to a conclusion. And so what I really see as a really neat opportunity is starting to build more of this. There are many resources that were pointed to, but I think there's a lot of discernment in, which ones are further along or less further along, and how do you define not only how to classify the hazard, but, which ones are higher confidence versus lower confidence. And so I think, some of my questions back to the panel is also, in moving in this direction, how do we start to develop those guidance and resources to help people select? And I think, Amy, you pointed to some great resources from PETA that we can start to think about in terms of incorporating these test methods. But again, just more thinking around how do we really give guidance to folks to integrate these to make consistent and appropriate classifications and confidence, so that we're really understanding and selecting the right chemicals. And the other thing I'll say, and Amy really spoke to this as well, is that, these different methods could, how you're going to use the results impacts which of these methodologies you choose. And, these green screen assessments and alternative assessments can be used on the front end in identifying, new chemicals in product development all the way through to evaluating products. And so I think that's a really important piece as well, to start understanding which ones can be used in what ways.
Mm-hmm. Those are great points, Sherry. And we've still got 11 minutes to go, folks. We want to hear from Lauren. So what are your thoughts and reflections, Lauren, based on the presentations and Sherry's insight thus far?
Yes, yeah, I'm thrilled with this webinar. I know experts like Pam and Amy have been working for many years on these methods, and it's very exciting to see the increased momentum towards their understanding and also the processes for getting them established and validated. So I'll just step back and say that Chem Forward is an online platform designed to scale access to chemical hazard assessments of safer alternatives in a centralized repository. Basically, the same information, data, can be used to report classifications, whether that be in GHS or Cradle to Cradle or Safer Choice or Green Screen. It's all based on the same information. And as Sherry mentioned, assessors have been using model data all along in tools like Green Screen, which I led for a number of years, and then primarily in areas like aquatic toxicity and persistence and bioaccumulation potential. And they're typically treated as lower confidence. However, sometimes the NAMs can be used as part of the weight of evidence when there may be conflicting data, say, between, say, a human study and an animal study for skin sensitization. That's a really great opportunity to dive into the mode of action and really understand the key steps and maybe support a decision one way or another. Is it or is it not a skin sensitizer? And I'm really grateful to Pam and Amy for driving home the point that NAMs are really important because they reduce costs, animals use, time to understanding. And you made a really great point that you're not creating NAMs to compete with animal models. You're creating NAMs to better understand the impact on human toxicity. So, and the fact that the skin sensitization NAM can outperform an animal model is really, really important. And I think you also did a great job making the distinction between screening chemicals for development or screening chemicals as candidates to use in a formulation versus bringing a chemical to the market through a regulatory process. So similarly, I'm really interested in which NAMs are ready for use now and for which endpoints. And I think one takeaway for me is the skin sensitization has a lot of credibility, in part based on the fact that it was developed with human data, human knowledge of human impacts. And that seems to me one that's ready to go. And the PETA resource is a wonderful resource for really deciding where to go when perhaps there are data gaps or there's a lot of uncertainty. So I think the questions that come up to me are what controls are in place to make sure that that, as Amy said, there's no one size fits all. You can't necessarily use NAMs for all endpoints. And I think, Amy, you touched a little bit on the process of consensus, of engaging diverse stakeholders in the development and establishment of methods. But what controls are in place to make sure that NAMs aren't used prematurely, whether for screening or regulatory purposes? I think that's a concern that people have. As Pam mentioned, you don't want them, they're fast, they're cheaper, they're less animal intensive, but will they give the health protection that is needed? And how can you sort of reassure and guide this development to ensure that we're really moving to a much better system than relying on animal tests that are simply another kind of model.
Great, thanks, Lauren. Let's move on to the next slide, and we're going to take questions from attendees. And here's the first one, and it kind of goes in line with what Lauren was just saying. What has been the level of acceptance of these methods with regulators so far? So I think Amy and Pam, if we can just hear from you, each of you, and obviously we don't want people to run out and let's say find a NAM and then take it to an agency and the agency says, oh, sorry, this doesn't work. What's your experience, the two of you? You want to start, Pam?
Yeah, I can start. And Amy, you'll probably touch on this. I mean, there's been a lot of activity through the NICEDM group to look at information needs and to start to say what information drives regulatory decisions. And the EPA Pesticide Program Office has been very active in really taking the lead on this under Analogue. And so they've been looking at deep drills on the data that they get for pesticide formulations and how that data has contributed or not contributed to the decision making. And as part of that, you know, the Pesticide Program Office has now said, well, you could waive dermal toxicity studies for the formulations because that often doesn't drive the data needs. I think that as we look at some of the more prioritization type of activities, they're more widely accepted, and that, you know, the point where we're going to start to say, we're going to use this specific assay to define a point of departure for a risk assessment, we're not there yet. I mean, if the case study with Syngenta and the inhalation NAM goes through, I think that'll probably be the first case where that's been done. So, I mean, we're moving in that direction, but it's a slow process, and we've got to decide how we can move that forward faster. So, Amy, you want to comment? I think you probably have a lot more pulse on that.
Yeah, that's a great answer, Pam. And I would just add that the burden really, I think, is on the company for early engagement with the agency. So there are various agencies, EPA, the FDA, and others that specifically say, come talk to us. They encourage their stakeholders to engage with them. And the Syngenta example that I gave is a good one, because that was the result of Syngenta approaching the agency saying, you know, we think we have a better way to do this. Can we talk through, you know, the approach that we've developed and get that to the place it needs to be to meet the agency's needs?
Yeah, that's great. Here's another question. So it was all the way back in 1959 that Russell and Birch published the Principles of Humane Experimental Technique, which really started the whole concept of 3 R's: replacement, reduction, and refinement. And it seems like for many years, I don't want to say it was senescent, but I can't open an email without seeing NAMs. Do the two of you, Amy and Pam, expect to see this great continued exponential rollout of NAMs? Because it just seems like over the past 5 years, it's really, really expanded. And I don't know if you feel like that, but that's my impression.
Amy, I'll let you go first this time.
Sure. Yeah, so it's a funny question. The concept of the 3 R's or NAMs is not new. And so I think it's been getting a lot of media attention lately. But I mean, this really, I mean, a lot of groups were working on NAMs before NAMs was even a catchphrase. So I do think, I mean, the science has advanced so much in the past decades. And I think what we're realizing now is that hasn't been translated into updating guidance and regulatory policies. So what we need now is really that roadmap, that plan to take the current state of the science and find a way to balance showing reliability and accuracy of these new methods while also not withholding these potentially life-saving methods that are going to be able to better protect human health and the environment.
That's great. In the interest of time, we just have a couple more slides that go into some resources. So let's move on, and we also have a quick poll. Let's move on to the next slide. So they're just in terms of what are additional resources. Some of you may be interested in presenting an abstract at the 11th World Congress on alternatives in animal use in the life sciences. This year it'll be in the Netherlands, and right now abstract submission and registration is open. So I know a number of us are attending that. And then next slide. And we are going to have another symposium, an international symposium on AA. So it's planned for the fall of 2020 on the west coast of the US in California. So stay tuned for A4's official announcement with the exact date, but please put that on your calendars. And finally, we want you to join the A4. In order for us to continue the advancement of safer alternatives and making it technically really sound, we need your participation. So join and become part of our collaboration. Next slide. And then we're going to go into a quick poll. So this will help inform us. Would you be interested to participate in an A4 discussion group on NAMs to further discuss additional issues and questions? Organizers and panelists do not vote. We want to hear if you're interested. Are you not at all interested? But please vote and tell us if you'd like to be part of a discussion group. NAMs are just like risk assessment, let's say, or any other thing. You really need to be trained by a community, and you need to see examples of well-done NAMs and probably NAMs that aren't so well done. Mm-hmm. So let's see what the results are like.
Okay, and Meg, if I could add, this is Lauren. We recognize that we have such great expertise and rich content on this webinar, and there's never enough time for questions and discussion. So this is an opportunity, if you're interested, to follow up specifically on the content of this webinar with more questions and discussion.
That's great. So obviously there's interest. So that was what we wanted to see, but we didn't want to cook the books. Let's go. So we'll put this session on the A4 website. I'd like to thank the panelists and the respondents. This has been a really great webinar, lots of content and lots of learning. So thank you everyone for attending. We appreciate your time. We know it's limited, and we look forward to seeing you at the International Symposium in the fall. Thank you very much. Goodbye.